I'm Tina Hessman-Say. I'm the molecular biology reporter and senior writer at Science News magazine. And I'm here today with Constance Hilliard.
I'm a professor at the University of North Texas in Denton, Texas. And my area is evolutionary history.
Right. It's been almost 20 years since scientists unveiled a rough draft of the human genome, but you say that calling this a human genome is a misnomer. Why is that?
What we now realize we have is essentially a European genome. Initially, when this Human Genome Project was first launched, scientists believe that, well, there are about 1.1 million gene variants in addition to the actual number of genes that we have as human beings. But time has gone by and now we know, no, it's not 1.1 million, it's 324 million. And not only that, but the 324 million is carried by the ancestral population, which is the population living in Africa. So the whole setup is a kind of one size fits all setup, but the one size is not universal. It's not even average. It's European, without there being a recognition of the fact that that will benefit people of European ancestry. But for other populations, we're going to miss many disease problems that are in fact creating health disparities between various groups, but we're only looking at it from such a narrowly defined perspective. And the perspective is one in which we assume that people of European ancestry are the norm, and we can therefore universalize their gene variants and their reference genome. I think the scientific community is definitely coming to a realization that the genome our genome is European and it needs to be fixed. But even the approach to fixing it sort of goes back to these kind of broad generalizations about race and so forth that really don't work. There are other countries that have finally woken up to the problem. And you look at a country like China. At this particular point, the Chinese began to realize that the reference genome is European. It's not helping their people. And not only that, but they took very concrete steps to stratify their genome. They've divided the population and therefore created a reference genome for Southern Han Chinese and a different reference genome for Northern Han Chinese and a different reference genome for Beijing Han Chinese and a different one for Koreans and so forth. Why are they doing that? They're doing it because otherwise it doesn't work. It only works for the population that you actually have all of the necessary data for. But I think maybe one of the problems is that it's very difficult to talk about race. I mean, there's no really safe space in the United States to really grapple with racial issues. And because of that, the scientific community backs away from it. But in backing away from it, it's not really using the precision tools that it has available to help other populations. And we're not going to be accomplishing our goals if we don't begin to stratify populations. But in order to do that, it's challenging because we have to stop talking about race and we have to stop talking about colorblindness. We have to start talking about the very particular ecological environments that populations come from. It's a matter of kind of raising our levels of consciousness and taking away some of the barriers. And the barriers are, a kind of fearfulness in dealing with anything that relates to differences in people. And we have to give ourselves permission to be able to delineate populations in ways that can really help them medically, as opposed to look good politically or something.